Namaste students, खुश रहो बेटा. I, JVN Dr. Chandrakala, Assistant Professor, Jyoti Vidyapeeth, Women's University, Jaipur. In today's session, we are going to discuss about the plant tissue culture. Plant tissue culture is an in vitro cultivation of plant cells or a tissue under an aseptic and controlled environment condition in liquid or on a semi-solid, well-defined nutrient medium for the production of the primary and secondary metabolite or to regenerate plants. In other words, it is an experimental technique in which a mass of cell or a callus is produced from an explant tissue. The callus produced through this process can be utilized directly to regenerate the plant heads or to extract or manipulate for a primary and a secondary metabolite. The plant tissue culture refers to the cultivation of a plant cells with normally formed of a multicellular tissues. So when a cell or a tissue is grown in an agar medium, the tissue forms a callus or a mass of undifferentiated cells. So the technique of the cell culture is convenient from starting and maintaining a cell line as well as to studying pretending to the organogenesis and the meristem culture. The technique of in vitro cultivations of the plant cells or the organ is primarily solves the two basic problems. Number one, micro-free uh, the plant cells or organ to develop into a microbial free or to ensure the desirable development in a cell of an organ by providing a suitable nutrient medium and other environmental conditions. So here the advantages of tissue cultures are availability of like raw materials. Sometimes the plant it is very difficult to cultivate in a certain climatic condition or a certain environment. So it to form a regular or a uniformity or availability of a plant in certain area. So this may be a helpful technique for the development of certain medicines from a certain plant, which may not be suitable for that particular climatic condition, but still can be developed into a plant culture. Fluctuation in supply and quantity or the method of production of food drugs in variable in or quality of change in depending upon the climate or the yield depending upon the climatic conditions can be studied and utilized with the plant tissue culture or new methods for isolation. This may be possible for the plants or obtaining certain plant part or extract from uh, through the process of plant tissue culture. So biotransformation can also be done where the plants or can functional groups or certain organic compounds can be modified by using a living cell or a plant tissue culture. So a disease-free or a desired properties can be obtained or a synthetic pathway can be traced by using this technique or immobilization of a cells where the plant tissue culture can be used for the plant preservation by immobilization of therapy or the continuous or uniform biomass can be obtained or medicinally important compounds can be synthesized using this technique or improvement of a medicinal plant species or propagation of plants without using a seed in a controlled condition. The disadvantage here is it is high level of expertization is required in this. A small error may lead to the complete collapse of the production or a plant unit or a lot of chemicals are required for the plant tissue culture which must be with high purity. There is no chance for the evaluation of the mutation. Culture on artificial medium may lead to the depression of unusual metabolic pathway or may be not beneficial for the biotechnologist. It may, in many cases, the secondary metabolite produced is negligible amount, which is of no use. So the protocol for individual plant differ with widely range on change in the medium or growth constitution, environmental parameters which affects the rate of the cell growth, accumulation or a secondary metabolite. To minimize the cell mass production or a cell suspension culture eventually becomes more dense or these present the problems of even aeration. So the expensive process aseptic condition has to be maintained throughout the plant growth. So it has a lot of limitations also when it comes to the point of limitations. Despite of that, it has a lot. It has a positive impact also. Thank you. This session is powered by 2.0